And welcome to this GCSE revision video, and this term we are focusing on required practicals. The required practicals will make up a significant part of your GCSE, so if you want to achieve good grades or improve your grades, they are a key thing to revise. Many students do not do and limit their own success. So today we're going to be looking at required practical 1, which is microscopy, which you probably did right at the start of your GCSE. So there needs to be key things that you need to learn. So you do need to know the key parts of the microscope, not necessarily every single part that is labelled on this diagram. So please pause the video and see whether you could label these and then move on to find out the correct answer. Key things that you need to know. It is very unlikely in GCSE that you'll be asked to label the tube. It is very unlikely you'll be asked to label the mirror. In fact, it'll probably be a light providing the light, funnily enough, through the stage. But the other ones you should know. So you should know this is called the eyepiece lens. This is called the objective lens, and normally there's 3 of those. And you have a coarse focus and a fine focus to focus that image for you. You need to know what the different roles and different parts of the microscope are. So you should be able to look at these functions and identify them with a part. So the part that would you would look through the sample, and often is times 10, is going to be the eyepiece lens. Okay, 3 to choose from, and the smallest is the easiest to focus, and that's one you use first, which we'll come on to why later on, is the objective lens. Holds the sample securely on using 2 metal clips is going to be the stages. Okay, this is usually placed in a drop of water on the microscope slide. That is your actual specimen. The flex lights upwards, that's going to be a mirror, and this quickly and easily moves the stage up and down to focus the sample. That's going to be your coarse focus before the fine focus slowly moves the stage to make the image very sharp. So you should be able to identify the roles of those. Probably unlikely that you'll be asked for many of the roles, but you might have to describe them in a method, and naming parts of the microscope is essential if you're asked that question. So you could be asked how to calculate the total magnification of the microscope. So this is not calculating the magnification of an image, which we'll come on to in a moment. This is calculating the magnification being used. So how you do this is you do the eyepiece magnification multiplied by the objective lens magnification. So if the eyepiece is 5 and the magnification of the objective lens is 10, you've got a total magnification of 50. This one you would have to do some division, so you do 200 divided by 40. Okay, and this one you do 400 divided by. Could be asked about magnification on a microscope image, and that is more likely. In fact, I don't think I've ever seen a GCSE question where a paper where this hasn't come up, regardless of tier or whether you're doing combined or separate biology. So if you learn the I-am equation, so image actual magnification, this gives you the definition of magnification, because magnification is the number of times bigger the image is compared to the actual size. You must say image and actual or real size in your answer. So magnification, number of times larger an image is compared to the real size of an object, and you do image divided by actual. And if I want to work out the actual size, I might do image divided by magnification. You're unlikely to be asked to work out the image size because you could measure that in the exam. And resolution is a term you need to know. Resolution is the ability to distinguish 2 objects close together rather than see them as a single object or a blur. So it's that idea of having a sharp image where you can see separate objects that are very close together. And if you get a blurry image in the exam, it's because the resolution is bad. If it's a small image, it's because the magnification is bad. Let's have a look at an exam question. And remember, in most questions, you are not given the equation. So if we have a look at this one together and work through it, we have got to calculate magnification. So straight away, sorry, my pen isn't working, bear with me. Magnification is image over actual. So therefore my image size, okay, if I look at this, it says the real length is 0.06, so that's my actual, so I'm going to do something over 0.06. And then if you look here, you've then got to measure the image size. Okay, now obviously you doing this on the computer isn't going to be particularly useful, but let's imagine that the image size, if you measure that in an exam, it is 5.7 centimeters or 57 millimeters. Okay, because they're the same units, they're both millimeters, you don't have to do anything, so therefore you would just do the sum 57 divided by 0.06, which would give me a magnification of 950, and just put a multiplication sign. See in a diagram or an image, sorry, that is from a microscope slide, well, the likelihood is it will be based on onion cells, which is what you would have used, and the image is something like this. So you can see here on the lower magnification, your diagram, that would probably be the best you could see. And if you were to look really close, you might get to see some of those things. But how do we represent? We have to draw scientific drawings, and scientific drawings have the following rules. You should only use a pencil. There should be no shading, so you can see here they haven't shaded anything, so it's a good diagram. There should be ruled horizontal lines for labels. There's no labels on this one, so that's one way they can improve. There should be no crossing lines for the label lines. The magnification should be stated, which this one isn't. The drawing should be titled to show what it is, and there should be singular lines, which this diagram has got. So this diagram is good for the drawing, but not for the labels. No magnification, no title. Scientific drawings then of a cell. So this one has magnification, the other one doesn't. It has lines that are drawn to a ruler labeling it just to the side, whereas this one has got lines with ruler but crossing over, which is not allowed as a result. So you might get given a exam question with the method, which means you won't have to describe the method yourself, or you might have to write it down. So I thought I'd show you this because it shows you the detail they would give you, which therefore you'd accept is the detail back. So the first thing this says is cut an onion into pieces using a sharp knife. Pull a thin layer off using a from one piece. So you need to think about why you do these things. So the questions could be, well, why would you do this? So the reason you would use a thin layer is so the thin layer is thin enough so the light can penetrate through so you can actually see the object. Place the layers onto a microscope slide. You might also want it with one cell thick as well there, I should say, so you're only seeing one cell. But really the light one's one you want to go with. Place a layer on a microscope slide, add 3 drops of iodine or other stain in different situations to the cells. Why would you do this? Well, the key answer that people often put is they put so you can see the cell, and you will not get the mark for that. What this does is it provides something called contrast. So the reason you can see the nucleus is the nucleus stains, the rest of the cell doesn't, so it's contrasting with the rest of the cell. And what this allows you to do is see more details and see different organelles in the cell. You would then apply very gently and then press down slightly a cover slip, and you put it down at an angle. One of the reasons is to remove air bubbles, which would be called artifacts, which means you can't see what you're actually looking for in there. Okay, and then you obviously place a slide on a microscope. You can see the different stages. So take the onion, use some tweezers, make the smallest layer as you can. And as I said before, just to really re-emphasize it with you now, why we're doing that. We're doing that so that you can see light passing through. Place the onion side on a slide. Okay. Make sure the onion is flat and does not have any folds. Take the iodine and add it on. And this is so you can see the organelles because it provides a contrast in colour. Gently press the cover slip down to make sure there are no air bubbles. Okay, and then you place it on the slide, and first of all you use the lowest magnification. Now the reason you use the lowest magnification is because you have what we call a wider field of view to start off with, and then you focus in on using the higher magnification of the objective lens. So you always use the smallest objective lens first with the lowest magnification because it has a wider field of view. And then you would use the coarse focus, and you would use that in the fine focus to bring it into focus for your eyes. So if you were asked to put the following statements in order, this would be not likely to ask an exam question, but if you press pause now and see if you can do it. Now you could potentially argue these slightly different order, but let's have a look at them. So the first thing I'd suggest is that you place the object on the slide. Okay. Second thing I would suggest is that you rotate the objective lens so it's at the lowest magnification. You then adjust the mirror, turn on the light. Okay. You would then need to look into the lens so you can actually see what's going on. You would then turn the coarse focus knob to bring it into focus. You would then do the fine focus, and then you turn the objective lens up if you needed to go onto higher magnification. So that is the key things you need to know about this practical. You need to be able to talk through the method. You definitely will be asked to calculate magnification, and you might be asked for reasons for doing different parts of the practical, and you may well be asked to draw scientific diagrams of photographs in the exam to do with cells and label them correctly.